and welcome to the podcast of Loughborough University's Institute of Advanced Studies, a podcast where our international fellows discuss the most pressing issues of today that we're all facing and share their exciting research ideas. We hope you enjoyed today's talk. Thank you for tuning in. Good afternoon, everyone, and welcome to the International House. I'm Professor Ksenia Chmutin. I'm Director of the Institute of Advanced Studies. Great to see so many people in the room. Welcome to those of you online. As always, International House is the place where we have the most exciting discussions on campus. It's always wonderful to have these discussions, and welcome to Alberto, who is joining us today. It's wonderful to host you. Couple of housekeeping rules. This is a hybrid seminar. We are recording the presentation. And the presentation will be published on our YouTube channel and as a podcast. Magically will appear in a couple of days. Just happened. We are not recording Q&A though, so if there's any discussions, any questions, that part will not be online. For those of you online, please post your questions in the chat. We are monitoring the chat, and of course, those of you in the room, there will be time for Q&A, but also we'll then continue having discussion over lunch. Over to Kate, who is hosting Alberto today together with Paul Wood. So Kate, over to you.
Perfect. So it's great to see so many people here. We weren't expecting how many people will be interested in fine sediment, so it's really good to see so many. We're delighted to host Alberto. So for those of you that don't know, I'll just start with a bit of applied history. So Alberto received his bachelor's degree in natural sciences, and then he went on to do a master's in environmental biology at the University of Turin. And this is where he sort of forged his interest in fine sediment, and I think he'll talk about that today. He then continued his studies doing a PhD in Turin, where he looked in more detail at the effects of hydro-morphological alterations, particularly siltation and flow intermittency on macroinvertebrates. And then since 2021, he's been employed as a researcher at the University of Eastern Piedmont, and here he delivers on the ecology module as well as the environmental impact assessment module. And when I was thinking about the introduction, I tried to remember the first time that I made contact with Alberto. So I think it was in 2019 when I was writing my FLF program, where we were trying to look at the impacts of the abiotic and biotic controls, including fine sediment, and I reached out to Alberto. And since then, we've worked on a number of projects together. So the first being combining both our PhD data, actually looking at a cross-country comparison of the effects of fine sediment. And then the Loughborough Lab team was lucky enough that we got to actually visit Alberto in Astana, which hopefully he'll show some pictures today. So in his setting, it's a really nice Alpine setting. So I think Alberto probably got the short end of the straw because there's no lovely scenery here in Loughborough. But hopefully there's an equally warm welcome. So I'll pass over to Alberto to talk us through some of his interesting research.
Thank you. Thank you very much, Kate, for this nice introduction. Hi, everyone. Thank you for coming. First of all, I'm very grateful to Institute of Advanced Studies and also to Kate and Paul for inviting me. This is my first time in UK. I'm very happy to be here. And today I'm presenting you some results of my research activity dealing with siltation in river. Before starting, let me to give you a brief introduction of myself, as Kate did before. I am Alberto Doretto. I work as a researcher at the University of Eastern Piedmont in Italy, and basically I am a river ecologist. My research activities is focused on the effects of hydro-morphological alterations in rivers, and in particular the impacts of these alterations on benthic macroinvertebrate communities for biodiversity and also for river biomonitoring. In this presentation, at first, I will provide you a brief introduction to siltation, why it is important to study this phenomenon. Then I will show you some case studies I carried out on this topic, and also some possible applications for river biomonitoring. Finally, I will conclude showing you some ongoing collaborations and other similar activities. Well, Let's start about siltation. Siltation is defined as the excessive accumulation and delivery of fine sediment in rivers. Siltation is usually recognized as one of the main hydro-morphological alterations around the world, concerning not only freshwater ecologists like me, but also local stakeholders like local agencies and associations. Here you can see some examples. Usually, fine sediment such as silt, sand, and clay is a typical component of the fluvial sediment and substrate. But there are some anthropogenic activities, for example, mining activities, deforestation, agriculture, road construction, and especially dam and its associated sediment flushing operations that can generate an excessive transport and accumulation of fine sediment in rivers. And this can result in an important environmental impact. Fine sediment in rivers can act as a disturbance in two different ways. For example, suspended sediments usually increase the water turbidity, and this can be a stress for some aquatic organisms. On the other hand, settled sediments can clog the interstitial spaces in the substrate. And again, this can be an impact for the aquatic biota. Both settled and suspended sediments can have direct and indirect impacts on aquatic organisms. To give you an idea, I added in this slide a graph from a publication. For example, fine sediment can play a direct effect on aquatic organisms in terms of the burial of animal, especially for the benthic organisms that live in the substrate. Fine sediment also reduce the habitat heterogeneity of a riverbed. And also fine sediment is a source of stress for aquatic organisms in terms of body abrasion and the clogging of some anatomical parts, for example, mouth parts and gills. Among the indirect impacts, we can mention the depletion of oxygen. Fine sediment usually alter the gas exchanges between the river bottom and the water column. And also the massive deposition of fine sediment reduce the availability of the main food resources for riverine organisms, for example, benthic algae and organic detritus. As a consequence, siltation can have a lot of impacts on all the biological components of the river ecosystem, from the food sources until the top predators. But in particular, benthic macroinvertebrates there are several different aquatic insects and other invertebrates are excellent bioindicators to study the impacts of siltation, because they are strongly related to the substrate and sediment condition. They are widely used as a bioindicator for assessing the ecological status of rivers around the world. And in particular, they are very useful to evaluate siltation. Most of all the results I will show you in the next slide refers to the response of benthic macroinvertebrate to fine sediment in rivers, and in particular in Alpine rivers, where I live. Well, after this brief introduction to siltation, now I show you some case studies I carried out during my academic career. The first case study, it was really the first experiment I carried out on this topic when I was a master degree student. So you can see how it started. And this study was carried out on two different alpine streams. The Lucerna streams it is characterized by a lot of mining activities in its watershed. By contrast, the Lucerna, the Combalusa stream was selected as a control stream. It is located a few kilometers apart from the Lucerna stream, and it is not characterized by mining activities, so in our study was selected as a control stream. The general aim of this study was to compare the biodiversity of benthic macroinvertebrate communities between these two alpine streams. In particular, in this case study, we adopted a manipulative experimental approach. At first, we placed different sediment traps in all the sampling stations. Sediment traps consist of a small plastic bucket with a metal grid inside. We also put some small stone on the top, and the traps were placed in the substrate of the rivers flush with the bottom. These devices allowed us to collect quantitative samples of fine sediment. The traps were placed on the same date in all the sampling stations and removed after 2 weeks. When we removed them, all the sediment, fine sediment inside them was transferred into a plastic tin and returned in a laboratory where the sediment was dried and analyzed according to the grain size classes. When we removed the traps, we also collected benthic macroinvertebrates as close as possible to the spots where the traps were placed, in order to rely on very similar biological and sediment data. Also, we collected in the field data on diatoms and benthic algae. We found that both the amount of benthic algae and coarse particulate organic matter was strongly reduced in the stream affected by siltation than the control stream. So these results confirm that siltation reduced the availability of the main food resources for riverine organisms. And when looking at the macroinvertebrate community, as expected, we found that both taxon richness and total abundance of macroinvertebrate were lower in the stream affected by siltation than in the control stream. But the most interesting results came from the analysis of the feeding functional groups. We were able to classify macroinvertebrates according to their feeding strategies. For example, filter feeders, shredders, scrapers, predators. And in particular, we found that fine sediment was an environmental filter that selected the composition of macroinvertebrate communities. In particular, shredders and scrapers. These categories include macroinvertebrates that feed on CPOM and benthic algae, respectively. The abundance of these groups was strongly reduced in the stream affected by siltation. During my career, I had the opportunity to continue my study on this topic, and in particular, as Kate mentioned before, we started a collaboration with the general aim of combining our results. In particular, in this case study, we tried to analyze our data to find if macroinvertebrates respond in a similar manner across different environmental settings. We are aware that siltation is an important hydro-morphological alteration around the world, but do macroinvertebrates respond in a similar manner, for example, in Alpine stream and in UK lowland rivers? That was the main goal of this study. In particular, in this study, again, we adopted a manipulative experimental design. We created some colonization devices, for example, small cages or cylinder. Some of them were filled only with gravel. They were the clean substrate, and some of them were filled with gravel and also fine sediment, the clogged substrate. All the colonization devices were placed in the rivers and then collected over different time scales, short, medium, and long term. Our results show that fine sediment altered the composition and diversity of the macroinvertebrate communities in both alpine and lowland rivers. This means that the taxonomic composition, the diversity of the macroinvertebrate communities associated with clear and clogged substrate was different. In this case, alpine and lowland rivers were similar, but we also found that the main mechanism that was responsible for these compositional differences differed from alpine to lowland rivers. In particular, in alpine streams, the compositional differences were mostly due to the nested scenario. It means that the macroinvertebrate community associated with a clogged substrate were a small and less diverse subset of the macroinvertebrate communities of clean substrate. By contrast, in the lowland UK rivers, both the nestedness and also the turnover, that is the species replacement, contributed to the compositional differences observed between clean and clogged substrates. So we also found some differences in the mechanisms that drive the response of macroinvertebrates to fine sediment condition between alpine and UK rivers. Well, the previous studies showed us that macroinvertebrates respond to fine sediment accumulation in rivers, and also that there are some differences according to the geographical setting. The next step in our analysis was related to river biomonitoring. Can we use these results to implement our river biomonitoring techniques? Yes, we can, but one of the most interesting aspects is that despite the negative effects associated with siltation and fine sediment in rivers are very known in scientific literature, specific biomonitoring indices based on macroinvertebrates have been developed only in few countries around the world. I'm aware that here in UK you have the PSI index. Similar indices already exist in the USA and also in Germany. All these indices have strong and statistical basis. They are effective in monitoring siltation, but they can have also some limitation. For example, I'm Italian and I am interested to apply these indices to my own study area, but there are some limitations. For example, the taxonomic identity of macroinvertebrate species can vary from one region to another one. Also, we I'm not sure that all the species respond in a similar manner. They have the same tolerance or sensitivity to fine sediment. And also another important aspect is that most of the biomonitoring indices I mentioned you before were developed in an agricultural context. And this can result in some possible co-occurring factors. In an agricultural landscape where siltation originates from the agricultural practices, maybe other co-occurring factors can have an importance, including chemical pollution due to pesticides, channelization, or the loss of riparian vegetation. To this end, some years ago, my colleagues and I tried to develop a new biomonitoring index based on the macroinvertebrate communities and also designed for Alpine rivers. Compared to the previous ones I mentioned you, this new biomonitoring index was a multimetric index. So we tried to identify what are the best community metrics that respond to fine sediment, and then to combine them into a new biomonitoring index. To do this, we first selected 12 different candidate metrics according to the scientific literature. Some of them are indices of biodiversity, for example, taxa richness, Ephemeroptera, Plecoptera, and Trichoptera richness. EPT richness refers to the most sensitive aquatic insects we can find in our rivers. But also we selected other different metrics, for example, some functional metrics related to feeding strategies or other important biological and ecological traits of macroinvertebrates. The performance of each candidate metric was tested and evaluated using previous data on siltation to evaluate the response of each of them with the amount of fine sediment. And based on our results, we found that the most sensitive metrics are three. Total taxon richness is a measure of the total biodiversity of the community, of the macroinvertebrate community. The EPT richness that refers to the most sensitive invertebrates in rivers, and also the abundance of ecological group A. This latter one is a functional classification of macroinvertebrates that includes those preferring fast-flowing waters and also large mineral substrates. According to our results, these are the most useful community metrics to assess the impact of siltation. And we try to combine them into a new biomonitoring index. We call it the siltation index for lotic ecosystem. This index varies from 0, the worst condition, the most impacted condition, to 1, the best condition or pristine condition. During my PhD program and my academic career, I had the opportunity to validate this new developed index in different new case studies, especially in relation to sediment flushing operation from dams. The first case study I show you was carried out in the Stura di Monte River. In January 2016, from 7 to 8 January, in a very short time period, a huge amount of fine sediment was released from the dam in the downstream section. I took the second picture in March 2016, a few kilometers downstream from the dam, 2 kilometers downstream, and as you can see, the water turbidity was still high due to the massive deposition of fine sediment. As a consequence, local authorities forced the owners of the dam to perform some ameliorative action. In May and June, on 2 different occasions, large volumes of water were released from the dam to remove the fine sediment from the downstream section in order to facilitate the habitat recover and also the recolonization of macroinvertebrates. Since we had quantitative data on macroinvertebrates in the same river section before the operation, we undertook a field study to monitor the temporal recovery of macroinvertebrates over time. And it was also an excellent occasion to test and validate our proposed index. When looking at the results, as expected, we found a strong reduction in the total tax richness and the total density of macroinvertebrates after the sediment flushing operation. Also, an additional reduction was observed after the two flash floods. But these operations were effective in removing fine sediment, and these facilitated the recolonization of macroinvertebrates. In figure C, you can see the performance of the STAR ICM index. This is the official biomonitoring index in Italy based on macroinvertebrates according to the European Water Framework Directive. But this is a generic index. It was not developed specifically for assessing siltation. As you can see, we found a change in the water quality class, but this mainly consisted in the shift from the high class to the good water quality class on the majority of the sampling dates. On the contrary, in figure D, you can see the temporal variation of our proposed siltest index. That was more correlated to the response of taxon richness and density. So these results illustrate that probably our stressor-specific index was more effective in monitor the impacts associated to sediment flushing operation and also the macroinvertebrate recovery. Then I had the possibility to validate again the index in other case studies, always in the Alpine areas and always related to sediment flushing operation. This case study originated from a collaboration with other Italian colleagues from the Lombardia region. And again, we monitored the impacts of a multi-annual flushing operation from the same dam, same river section, but year after year, different sediment flushing operation. Again, we found that these operations strongly altered the composition and diversity of macroinvertebrate communities, and especially these consisted in the loss of taxa during the first days after the operations. In particular, our proposed siltest index was strongly correlated to the nestedness, the main mechanism explaining the compositional variation of macroinvertebrate communities. And again, these results confirm that a stressor-specific index is probably more effective in monitor these type of operations. And this assume in particular importance, especially if you have a look at the Alps. Alps are a very important and beautiful area. I strongly recommend you to visit them if you have the possibility. Usually, the presence of a human in the Alps is very low. They are an important hotspot of biodiversity in Europe. But when you look at the rivers, damming and its associated sediment flushing operation is surely one of the most important human pressure, as you can see from this picture. In fact, in the last decades, the number of papers dealing with this topic increased over time, as you can see from this graph, and the majority of them assessed the impact on macroinvertebrates, along with other biological components like fish or other aspects. And also, when you look at the Italian situation, you can see that the temporal variation of the in the number of sediment flushing operation increased over time. In this graph, you can see the number of these operations among the different Alpine regions of Italy. You can see some differences from one region to another one, but in general, from 2001 to 2024, the total number increased. Again, as a demonstration that sediment flushing operations are a very strong and current challenge in the management and conservation of alpine rivers. And this is why I'm still working on this topic. In particular, since October 2023, I've been leading a research project dealing with sediment flushing operation in the Alps. This project includes river ecologists like me, but also hydraulic engineers. So the main goal of this project is to develop new biomonitoring methods for correctly assess the impacts associated with sediment flushing operation, and also, if possible, alternative strategies to manage the sediments in the Alps. Some of the results and case study I showed you before originated in the framework of the activities of this project. To conclude, what are the take-home message of this seminar? At first, benthic macroinvertebrates are excellent bioindicators, not only to assess the general ecological status of rivers, but especially for assessing the impacts of fine sediment, due to their strong relationship with the substrate and near bed conditions. Also, when looking at the macroinvertebrate community, we can identify the most sensitive metrics, and we can use this information to improve our biomonitoring techniques. In the last few slides, I would like to mention you another approach to study fine sediment in rivers. I refer in particular to the use of flume-based mesocosm. Mesocosm in an ecological study offer a lot of possibilities and advantages. For example, researchers are allowed to manipulate some parameters like sediment, substrate composition, flow, and in this way, we are able to perform some manipulative studies. As a member of the Alpine Research Center in Ostana, a small but beautiful village in the Italian Alps near the border with France, I have access to that facility, mesocons facility, you can see in the picture. These flumes are located in the left bank of the Po River, around 1,000 meters above the sea level. And the main reason why I tell you about this mesocle is that Paul, Kate, and I used these flumes in the last years. From July 2022 to May 2023, we carried out a joint experiment about siltation. So we had the opportunity to study together how macroinvertebrates respond to fine sediment addition. And you can see some pictures of our experiment in Ostana. The most important thing is that now we are analyzing our data, and in particular, Paul and Kate and their team carried out a similar experiment here in UK by using similar flumes. So the general aim is to combine our data again to evaluate if macroinvertebrates respond in a similar manner across different geographical settings. Well, I'm at the end of my presentation. Of course, I would like to thank again the Institute of Advanced Studies and Paul and Kate for inviting me, as well as all the other people that helped me during my study and the experiment I showed you. And obviously, thank you very much for your attention.
Thank you for listening to the podcast of Loughborough University's Institute of Advanced Studies. A quick reminder about how to stay connected. You can find us on Blue Sky, LinkedIn and Instagram, and you can also watch the presentations on YouTube. The podcast is available on all the major platforms. Please download, share, and most importantly, subscribe. See you next time.